Dear students, after having looked at how to score MS1, that is the mass, and the PSTs, that is the peptide sequence tags, and the in silico and in vitro spectral comparisons, we saw how can we integrate all of these three into a system. This search system still requires to combine the scores. So it's not just important to order these steps, but also the score that is obtained from each of these steps needs to be integrated. So in this module, we're going to look at an integrative scoring scheme that will help you to arrive at an overall score for each protein in the protein database. So as I just mentioned, there are three components to the scoring. The first is the molecular weight, the second is the PST, and the third is the in vitro and the in silico comparisons. Before we get into the scoring scheme, why is it necessary? It's a very important question. The score from the molecular weight, the score from the PST, and the score from the in vitro and in silico spectra is not normalized to a specific value. Score from the molecular weight will just tell you how close the experimental MS1 is to the candidate protein's mass from the database. So this can be a number ranging between a very small decimal fraction or a very large number. For PSTs, we looked at the length of the PSTs and how they compare with the candidate protein. So that can range from, let's say, integer 1, 2, 3, 4, and so on. For the in silico and in vitro spectral comparisons, we simply got the counts. How many peaks matched with the in silico spectra. So this information is quite heterogeneous, and we need to integrate that into a normalized score in order for each of these components to have a meaningful say towards the final score. So this is why this is an extremely important step in the protein search. Let's take a look at the options that we have. Simply, we can just add up the scores. So the score from the molecular weight can be added with the score from the PSTs to the score from the experimental and in silico or theoretical spectral comparison. But as I was just mentioning, you cannot add oranges and apples and bananas. So this is exactly like that, because the score from the molecular weight may vary between 0 to the molecular weight of the protein, which can be quite large. The score of the PSTs can be 0, 1, 2, and so on, till maybe typically 8 or 9. The score between experimental and theoretical spectra can actually vary between 0 to the length of the protein, or simply the number of amino acids. So these are three very different quantities. One cannot be normalized to another or to the third. So we need to really see how to combine them in a meaningful way. One step towards this meaningful approach would be to divide each score by the error. So in this way, you will have a normalized score with reference to its error. Here, you have the score for the PSTs, and you divide it by the root mean squared error, and you sum it over all the PSTs. Similarly, you compare the score from the experiment and theoretical spectral match, and you divide it by the error, overall error that was there during the matching. And you do it for the entire protein length. So, by adding all of these three quantities up, you can have a better score as compared to the score that we obtained earlier. Of course, we can keep on optimizing this entire scoring strategy and arrive at something that is extremely depictive of the score of the protein. In the commercial industry, there are complex non-linear functions that are used to score this MS1 and MS2 spectral comparison. For instance, in Mascot from matrix science, they use an extremely complex non-linear function, and mostly these highly proprietary software are not made public. So what you can do is you can design your own scoring scheme and you can contribute towards the research as well. So in conclusion, a composite scoring scheme is needed to combine all of these factors together into a homogeneous score, and the ability of a score is measured by the scoring system's ability to separate the true positives from the false positives. True positives being the correct matches and false positives being the incorrect matches. So if your scoring scheme can put them further apart, then you have a good scoring scheme with you.